Entomopathogenic fungi as a potential alternative to chemical pesticides. Today for this presentation, I will be covering current climate of pesticide use. What is entomopathogenic fungi? How does it work as a pesticide? How effective are entomopathogenic fungi at controlling pests? The Red Queen hypothesis. What are the long-term impacts? And the conclusion. The current climate of pesticide use. Pesticides are used to increase the yield and quality of crops, maintain the aesthetic of an area, and even to control vector-borne disease. Their use became large-scale in the 1940s after World War II. One of the most used pesticides was DDT. DDT, an organochloride pesticide, was very efficient at controlling mosquitoes, the vector for malaria. DDT was banned in most of the world in 1972 due to the negative effects on the environment and human health. To date, organophosphate pesticides are the most used type of insecticide and herbicide. They are used in agriculture, on golf courses, for wood processing, and even in the flea and tick collars for pets. As time has progressed since the start of large-scale pesticide use, many negative effects have been observed. When pesticides are applied, they do not stay only in that location. Through volatilization, runoff, and leaching into the soil, neighboring areas are affected by pesticide use. Once in the water, aquatic life see many side effects, such as erratic swimming, reduced fertility, and even death. Pesticides in the soil reduce the amount of soil microbes present, therefore reducing the quality of the soil. This leads to difficulty growing crops. Not only are aquatic animals and soil microbes affected, so are terrestrial animals. One of the most well-known examples of terrestrial animals being affected by pesticides is the effect of DDT on raptors. Because DDT is that soluble, it builds up in the tissues of animals. Because raptors are apex predators, they are consuming harmful levels of the pesticide that have built up in the tissues of their prey. This consumption led to these birds not being able to lay healthy eggs. The eggs that were laid had brittle shells that were regularly cracked before the chick was ready to hatch, resulting in death. The chicks that were able to hatch often died young because they were very unhealthy and consuming even more DDT. This led to many raptors, including the bald eagle, to become endangered. Human health is also affected by pesticides. Organophosphate pesticides work as acetylcholinesterase inhibitors in insects. These chemicals prevent the breakdown of acetylcholine molecules so that they are unable to be reabsorbed from synapses, causing constant activation of the receptors. This leads to paralysis and then death. The problem with this is that humans also have acetylcholine. Exposure to these pesticides cause the same sort of inhibition seen in insects. These negative effects have been seen by the public and have caused consumers to call for reform. Biopesticides are derived from natural sources that have fewer negative side effects. Entomopathic fungi are being explored as a promising alternative to chemical pesticides. What is entomopathogenic fungi? By breaking down the word entomopathogenic, we see that they are a type of fungi that infects insects. These fungi start by growing on the outside of the insect. Then by using cuticle-penetrating enzymes, the fungi then grow into the insect, entering their hemolymph fluid and killing them. After entering the hemolymph fluid, if conditions are favorable, the fungi will produce fruiting bodies and spread their spores. In this image, we see an insect, probably an ant, that has been infected by entomopathogenic fungi. This is characterized by the fluffy white coating as well as the fruiting bodies we see. How does this work as a pesticide? The cuticle penetrating enzyme is specific to insects. Only insects are going to be affected by the application of this solution. Different fungi have different cuticle penetrating enzymes, resulting in only the target insect being killed. A caveat to this, though, is that there is some overlap between the types of insect that an enzyme can be used on. This means that several different insects can be affected by the same enzyme. This figure is from an experiment that explored the potential of using L. leishmanii as a method to control ticks on cattle. The experiment exposed female ticks to a solution of L. leishmanii conidia and observed the effects for 10 days. It was found that this fungi is capable of infecting and killing ticks, but does not prevent the hatching of the eggs from the infected females. How effective are entomopathogenic fungi at controlling pests? When developing a pesticide, efficacy is important. The pesticide should work to kill bugs quickly and at a reasonable concentration. In a study performed by Rosman et al., 4 different species of entomopathogenic fungi, V. leishmanii, P. fumarosus, M. anoplosi, and M. anoplosi, were tested on cabbage aphids. They did 2 tests, one to find the most effective concentration of conidia per milliliter, and one to measure the mortality rates of different concentrations over a 7-day period. It was found that V. leishmanii was the most virulent, the optimal concentration being 1.88 times 10 to the 6 conidia per milliliter. Looking at the mortality rates for this concentration, between 75 and 91.52% of the aphids should have been killed. When comparing entomopathogenic fungi to chemical pesticides, a major drawback is the time required to kill the majority of insect pests. Chemical pesticides such as chlorpyrifos, an organophosphate pesticide, kills the majority of the pest in the first 24 hours following application. Entomopathogenic fungi, however, take up to a week to kill the majority. When looking at how entomopathogenic fungi work as a pesticide, we saw that they are only able to infect a narrow range of hosts. This is because the cuticle of the insect contains a range of different lipids and proteins. In order to infect the host, the fungi must be able to break through the cuticle. This requires cuticle penetrating enzymes. These enzymes are only able to target specific molecules in the cuticle, resulting in the fungi only being able to infect hosts that have this molecule. This image from Kehani shows a nonspecific example of how the fungi infects the host. This is beneficial to ensuring that only target pests and other insects that have the cuticle molecules are killed. Red Queen hypothesis. In Lewis Carroll's Alice in Wonderland, the Red Queen says, "Here you see, it takes all the running you can do to keep in the same place." Evolution is compared to the caucus race. In order to not fall behind, the species must adapt and evolve. Species that are competing for the same resources must continue to evolve and adapt just to maintain their relative fitness to each other. The relationship between the fungi and the host push each other to continue to adapt in order to maintain their relative fitness. This is also known as coevolution. The fungi involved have an increased virulence, or increased ability to infect and cause disease. This is beneficial for the fungi to infect more hosts. The fungi are also able to make infected hosts more attractive to other insects. They do this by causing the host to expose their abdomens, a very attractive trait. This allows for conidia from the infected host to transfer to the mate. Then this mate is then able to take the conidia and transfer them to another insect, resulting in an increased number of insects infected. In several insect species, it has been observed that infected hosts produce less eggs. This reduces the amount of offspring that will have been exposed to conidia. Reducing the amount of food the infected host consumes decreases its body size. This reduction in size slows the spread of the fungi into the vital tissues of the host, as well as provides the fungi with fewer nutrients to grow. Changes in the location of insect feeding are seen in the infected individuals. This behavior can broaden the dispersal of the conidia, but also can be an attempt to isolate and prevent further infection of the community. Similar to a fever in humans, increasing the body temperature of infected insects will slow the progression of the infection. While insects do not create their own body heat, they do this by physically moving themselves to a warmer location. What are the long-term impacts? Entomopathogenic fungi are naturally occurring in soil ecosystems. Different species of this fungi thrive in different soil conditions. Using entomopathogenic fungi as a pesticide has the potential to disrupt the soil ecosystem by introducing new fungi or by increasing the amount of an existing fungi. The health of the soil in the ecosystem is an important part of agriculture. Healthy soils will lead to crops that have a larger yield and better quality. This brings us to our conclusion. Currently, entomopathogenic fungi are not effective enough to be used at a large scale to replace chemical pesticides. The amount of pests that are left after application negates the benefits of switching to entomopathogenic fungi. As of today, it is incredibly difficult to manufacture entomopathogenic fungal pesticides. It is required to grow the fungi on a substrate, dry out the conidia, and then resuspend them in a solution. This process is lengthy and very costly. In the future, it is possible to have fungi that have been genetically altered to be more infectious. Research is currently being done to explore this possibility. In order for these fungi to be used at a large scale as a pesticide, manufacturing has to be bumped up to be able to handle this demand. Increasing the efficacy can only help so much if the pesticide cannot be manufactured at a scale large enough to get the pesticide to the farm for use. As of right now, chemical pesticides are the best option for pest control. But in the future, it is entirely possible for entomopathogenic fungal pesticides to be used large scale and to completely replace chemical pesticides.